Thanks again, Bob. Thanks, Sam.
Yeah, of course. So, good afternoon, everyone. I'm Sam Wood. I'm one of the internal medicine chief residents. So welcome to Grand Rounds, virtual. Next week on September 13th is the annual Kochman Lecture in Rheumatology. Dr. John Stone is going to be visiting us from MGH. He's going to be speaking on IG4 related disease, on which he is a world expert. So that'll be a really good one. Next week's Grand Rounds will be all virtual as well. And hopefully we will have our new AV setup ready to go the following week. So today's Grand Rounds is our quarterly ICTR session from the Institute for Clinical and Translational Research. The title is Discovering New Medicines in Academia, and we are going to be hearing from Professor Barbara Slusher today. Dr. Slusher is visiting us today from across town at Hopkins. She's a professor of neurology, neuroscience, as well as pharmacology, psychiatry, medicine, and oncology there. She serves as the director of the Hopkins Drug Discovery Program, the vice director of the Peterson Brain Science Institute, and the co-director of the Johns Hopkins Center for the Advancement of HIV Neurotherapeutics. She received her, excuse me, my screen share. Fix that again. There we go. She received her undergraduate degree in chemistry from Dickinson University and her PhD in pharmacology and molecular sciences from the Hopkins School of Medicine, simultaneously with a master's in administrative science from the School of Business there. Before joining the Hopkins Drug Discovery Program in 2010, she spent 18 years in the pharmaceutical industry and has extensive experience in drug discovery through early clinical development, and has been involved in the successful development and launch of several FDA-approved medicines. She's published over 300 scientific articles and is an inventor on over 100 patents and applications. At Hopkins, she leads the drug discovery program with a team of over 25 chemists, pharmacologists, toxicologists, pharmacokinetics experts, and others. They identify novel drug targets and translate them into new drug therapies for clinical development. Since joining Hopkins, she's also co-founded 4 new companies with over $150 million in financing, and she founded the first international consortium of academic drug discovery centers with over 150 university-led translational centers that bring together the growing community of drug discovery. So without any further ado, please welcome Dr. Slusher.
Thank you very much for that kind introduction, that long and kind introduction. Let me start to share. Okay, can you see my slides? Give me a thumbs up. Okay. All right. Well, first of all, thank you very much to Dr. Davis and others for inviting me today. I'm excited to be here. As Sam mentioned, I've spent my entire career doing drug discovery and development, the first half in pharma, and then more recently within academia. And what I'm going to talk about today is discovering the process of discovering new therapeutics within an academic medical center. I'm going to cover 3 topics. First, I'm going to start with telling you a little bit about the center that I run, this Johns Hopkins Drug Discovery. I'll talk about our capabilities, our services to the community, and our commercialization efforts. And the second topic I want to talk about is really the changing ecosystem of drug discovery. Over the past 20 years, there's been changes in pharma and NIH and academia, with academia playing really a rising role in the drug discovery arena. And so I'm going to go through some of those changes and updates. And then third, I'm going to spend, and this is the majority of the time, I'm going to give you some vignettes or examples of academic drug discovery projects, things that we discover in academia and ultimately we're able to spin out and to commercialization. So that's where I'm headed. Let's first start with the Johns Hopkins Drug Discovery. So in 2010, a drug discovery team from a company called MGI Pharma moved to Hopkins. Okay, so that's unusual. So how did that happen? So there was a company called MGI Pharma. We were in Boston and Baltimore and Minneapolis. We had 3 sites. I was the head of research and translational development for this company, and we were acquired by the big international pharmaceutical company Eisai for about $4 billion. So this was a very nice exit. Eisai wanted to move everybody, centralize everybody to Boston. At the same time, Hopkins was looking to recruit in a team of translational faculty. And so the long and short of it is a team of us, about 16 of us, moved from pharma into Hopkins to start this drug discovery and development program. And when we came to Hopkins, our new job basically was to work collaboratively with faculty and translate the discoveries into clinical therapeutics. And so we're a team of really small molecule focused drug discovery experts. We have a team led by Takashi Tsukamoto that does med chem. This is small molecules, chemical synthesis, structure activity relationship, hit to lead chemistry. We have a team that does assay development and high throughput screening to identify new leads to those targets. We have another team run by Dr. Rana Reis, who runs our drug metabolism and pharmacokinetics group, doing both in vitro and in vivo pharmacokinetics. Then lastly, we have a team led by Diane Peters that does in vivo pharmacology and toxicology with all kinds of routes of administration and formulation help, non-GLP tox, and several different animal models. So this looks very much similar to what you'd see as a research team in a pharmaceutical company. And over the last, since we've been here since 2010, and over the last 12, 13 years, we've prosecuted 12 different drug discovery projects. And I'm showing those projects here on the left. And they're in all kinds of different areas. So we've worked in brain cancer and antivirals, anti-inflammatory bowel disease, cognition, cancer, pulmonary. And this has really been opportunistic due to the projects that have been brought to us for doing drug discovery. But these efforts in these 12 drug discovery projects have led to multiple collaborations with pharma. In fact, 4 of these drug discovery projects have now been out-licensed to the pharmaceutical industry for development. In addition, we've spun out 4 new companies. We've raised about $150 million to move those projects forward. And so today, when I go in the last part of my talk, when I'm going to go through some of the examples, these vignettes of academic drug discovery programs, I'm going to talk about one that's focused on brain cancer that we've collaborated with Eisai. I'm going to talk about another project that led to the spin out of Drayson Pharmaceutical, which is focused in cancer on glutamate antagonist. And lastly, I'll give the example of a very new project focused on inflammatory bowel disease that we just partnered with the Bishadi Foundation here in Blackbird Labs here in Baltimore. And so I'll walk through those. Now, in addition to doing drug discovery and trying to commercialize these technologies, we also do a fair amount of services to the community here. And this is really how I met Dr. Davis and others at University of Maryland through the ICTR, collaborative ICTR. Our group is part of the Drug Discovery and Development, or D3 program. And through this, we provide on an annual basis about 150 to 200 services to the faculty. And when I say services, I mean things like help with drug discovery grants. We do design support for studies. We do a lot of assay development, initial screening assistance. We do pharmacokinetic sample analysis, chemical synthesis, a lot of consultation on how to do drug discovery and how to commercialize those. So these are a lot of the services that we provide to the faculty here. In addition, given our relationship with the University of Maryland, we've also several times have, in collaboration with Paul Shapiro and Jim Pauly at the pharmacy school, we've had several local drug discovery and development symposiums that we've co-directed, and I think that's led to much more interaction between the two institutions in the same town. So that's, I think, been very productive as well as enjoyable. Okay, so that was the first topic I wanted to cover, just to tell you a little bit about Johns Hopkins Drug Discovery, what we do, our capabilities, and our commercialization efforts. So now I'm going to move to the second topic, which is this changing ecosystem of drug discovery. So if you look over the last 20 years, there's been some really significant changes in those areas in drug discovery led by pharma NIH, as well as academia. And so I'd like to walk through those. All right, first, there's been significant changes within the pharmaceutical industry, which has really led to this rise of open innovation, where pharma is really enhancing their collaborations and sharing their risk. And I can't really overemphasize this enough. I worked in pharma during the 1990s and early 2000s. And when I was there, it was very much a closed innovation system. It was secretive. It was intramural. We didn't publish. We didn't present at meetings. Everything that we did that we were interested in was really focused on patents and really secretive and intramural in its focus. That's very different today in the pharmaceutical industry. There's been much more focus on open innovation, collaboration, and there's much more interaction with pharma, with small biotechs, with pharma, with academia, with pharma working with other pharma. So this has really been a significant change in a short period of time. Now, not only have there been changes in pharma, but there's also been changes in the NIH, where NIH has been increasing their interest and their efforts and their funding within drug discovery and translation. And I think this started initially under Elias Zerhouni, who started the NIH roadmap focused on translational research, which led to the ICTR and the Molecular Libraries Program, the RAID program, the CAN program. And more recently, under the leader Francis Collins, we started an entire new center at NIH focused on translational science, the NCATS Center, which really is focused on translational research, catalyzing public-private relationships, repurposing drugs, with really a focus on improving the translation of NIH-sponsored research into practice. And so this has also been, I think, a significant change within a small period of time. So changes in pharma, changes in NIH. The other area that's changed is academia. And there's been a fairly dramatic increase in academic medical centers getting involved in drug discovery or getting involved in translation. So here's a map of the US in 1990, and these little red dots were academic medical centers that had therapeutic focused programs, so drug discovery programs in them. That's 1990. If you look 20 years later, there's about 25 programs focused on translation in academic medical centers. And then if you look last year, which is the last time we counted, there's over 100 drug discovery programs at academic medical centers in the United States. So again, in a pretty short period of time, there's been a real dramatic increase in the drug discovery efforts within academic medical centers. And these data came from the Academic Drug Discovery Consortium. This is a consortium of academic centers that have come together to really to understand each other, understand the talents and expertise at the drug discovery area, so that it can enhance collaboration and interaction with pharma. And this is a consortium that I started about 10 years ago. We now have about 2,000 members and about 150 different centers that are members of the Academic Drug Discovery Consortium. So not only is academia have more centers, but those centers are seeing more productivity. And so if you look at the number of academic pharma partnerships, they are also very much on the rise over the past 2 decades. So this is looking at the number of executed deals between academia and pharma over the past 20 years. And you can see there is a dramatic increase, 16-fold increase over the past 20 years in the number of executed deals between academia and pharma. So this has been a big area on the rise. Not only is academia working more with pharma, but also there's been more investments in academic startups over this period of time as well. And I took this is the sources here below, but there's been about a 10-fold increase in the money investments in university startups over the past 10 years. And this is showing these investments in all areas: consumer, energy, media, service, telecom. But here in green is health. And you can see it's the exact same trajectory, very large increase in the investments being put into academic translational startups. So All right, so that was the second topic that I wanted to talk about, is that there's been a real rise in academia and drug discovery, changes in pharma, changes in NIH, and certainly changes in academia, where they're getting much more involved in translation, much more involved in spinning out companies and working with pharma along those lines. So academics' role in drug discovery has really changed. Okay, so that was the second topic that I wanted to cover. The third topic, and then where I'll spend most of my time today, is I'm going to give 3 examples of academic drug discovery projects, 3 vignettes, if you will, and I'll spend about 10 minutes on each. And each one's going to sort of have a lesson or an example of how the academic drug discovery programs can move forward. And so the first vignette is focused on inhibiting a transporter called System XC in the brain for the treatment of brain cancer. And I think this, as I go through this vignette, hopefully this is going to give you an example of how academia and pharma can innovatively partner for the better of, I think, drug discovery, and if successful, ultimately for patients. And so this is an example of our team at Johns Hopkins Drug Discovery working with the pharmaceutical company Eisai. So let me just give you a little bit of background. So this is focused on a transporter, actually an antiporter. Antiporter just means that it's got something, this transporter has something going in when it has, and also something going out. And this transporter is called System XC. It's a cysteine glutamate antiporter. You can see in this schematic, cysteine is transported in, which is then reduced to cysteine, which is involved in the synthesis of glutathione and ultimately redox control. But at the same time, cysteine's coming in, glutamate is being kicked out. And so this antiporter is involved also in the non-vesicular release of glutamate. Okay, so why are we interested in this? So over the past several decades, people have been interested in System XC because it's highly upregulated in brain cancer. And here's several publications that talk about System XC in brain cancer. So basically what happens, this is a normal cell. It's got System XC, cysteine comes in, makes glutathione, involved in redox, and glutamate goes out. Under normal conditions, the glutamate that is released from the cell is taken back in by the glutamate transporters, GLT1 and GLAS. So cysteine comes in, glutamate goes out, then glutamate is taken back in by these transporters. Okay. However, in glioma, the situation is changed, and it's changed because System XC is upregulated about 2 or 3-fold. And so again, you have more cysteine coming in, more glutamate going out. But the other change in brain cancer is that these glutamate transporters are significantly downregulated. And so what happens is you're getting cysteine in, more glutamate out, but less glutamate being taken back into the cell. And what this results in is more glutamate release, less glutamate uptake, which results in very high extracellular glutamate outside the tumor, which can lead to seizures and ultimately excitotoxic death to the surrounding tissue. And if you think about this from a cancer perspective, it's pretty smart, right? So cancers upregulate this System XC. So cysteine comes in, they make more glutathione, they can help with redox control, which is obviously important in rapidly growing tumors. At the same time, they're kicking out this glutamate and basically killing the surrounding cells so the tumor has room to grow. So from a cancer perspective, it makes sense that this System XC is upregulated. And so what's been shown is that there's been actually many different publications, several high-profile publications showing that if you inhibit this antiporter, if you inhibit System XC, either genetically or pharmacologically, you can decrease that peritumoral glutamate. You can decrease the seizures. You can decrease tumor volume and actually prolong survival. And so this is a summary of that taken from several Nature Medicine and neuroscience publications showing that in mice, if you inhibit system XC, you can inhibit this tumor-associated glutamate release, you can inhibit the number of seizures, you can decrease tumor volume and also increase survival. So if you're a mouse, inhibiting system XC is very beneficial to the brain cancer and the propensity for seizures. But this isn't just a mouse phenomenon. In Science Translational Medicine more recently, it was shown that in patients, if you have high levels of system XC in your glioma, it's associated with a higher number of seizures and a poorer prognosis. So patients that have high levels of this system XC die at about 9 months earlier than patients that have low levels of this system XC. They also were shown to have more seizure propensity with high system XC. So several years ago, we got interested in this as a drug discovery target. And we looked in the literature and there was nothing. There was no real good drugs. There were no potent, no selective system XC available. People were using these two drugs that I show here, sulfasalazine and 4-S4-CPG. And they're both lousy. They're both micromolar potency, so not very potent. Sulfasalazine itself is extensively metabolized here by azoreductase, and the metabolites are inactive. And S4-CPG is actually a better metabotropic glutamate blocker than it is system XC. In addition, they have these carboxylates in their structures, which makes it difficult to cross the blood-brain barrier. So basically, there really was no potent and selective inhibitors available. So our group got involved at this stage, and our first attempt was to try to take these molecules and modify them to make them more potent, make them more selective. And so I'm gonna not bore you with all of the chemistry details. I'm just gonna tell you that we failed. we made about 100 compounds on both of these scaffolds, and while we were able to change some stability, a little bit of potency, in general, we were not able to optimize either of those leads. So, but we were really still interested in this project, and so the next thing we did was we said, let's start at the beginning. Let's develop a high throughput screening assay and see if we can come up with some new leads or new structures that inhibit system XC. And so Camila Rojas and Ajit Thomas in the lab established the first high throughput screening assay for human system XC. And it was a fluorescence-based assay using these astrocytoma cells that express high levels of system XC. And it was actually nothing special. We put in cystine, and if you add cystine, you get glutamate kicked out, and then you can measure glutamate by a series of oxidation reduction reactions. This assay, though, was very well behaved. It had a Z prime at about 0.7, so it was real reproducible. And so then we had this assay. Then we said, okay, now what library of compounds are we going to screen? Right? So I came from pharma, and the pharmaceutical companies typically had millions of libraries that had millions of compounds that were pretty drug-like that we would use for screening. However, in an academic setting, we don't have that type of library. Certainly, you can purchase libraries, smaller libraries, but not the diversity, not the numbers that you can have in pharma. And so what we tried to do, and we're successful with AZI, was to try to find a pharmaceutical company that would collaborate with, that would allow us to use their compound library for screening this target. And so ultimately, we executed a high throughput screening agreement with AZI. Actually, I talked to many pharmaceutical companies and was unsuccessful, so I'm not showing those. But it was an effort. It was a long-term effort to try to find someone that would partner with us for this screening. A lot of words on this slide, but let me just explain how this works. We meet with AZI every year, and we share targets with them that we're interested in. If they're interested in the target, they tell us, yes, these are the ones we're interested in. We then, at Johns Hopkins Drug Discovery, develop a high throughput screening assay. If successful, we then transfer that assay to AZI. They screen their large compound library and then give us back the hits. And then we do drug discovery on those hits. And if successful, they have first rights to in-license the drug. And at least from my perspective, this is a total win-win. So from JHU, we don't have access to a large, diverse chemical library. Now we do. From a pharma perspective, they have now access to novel targets that they wouldn't otherwise have prosecuted. And so when we signed this this deal with AZI, it actually was listed that week in the pink sheets as one of the deals of the week, innovative ways that pharma and academia can work together. So this was we've done this screening collaboration with AZI multiple times, but this System XC was the first time that we ran this this partnership. And so just in general, this is how the partnership works. We disclose targets. The company then selects targets of interest. We then develop a high throughput screening assay. We then transfer that to the company, and the company screens their large chemical libraries. Those hits then come back to us for confirmation and orthogonal assay. If they hit, they then share the structures. And then either all by ourselves or in collaboration with the company, we then do drug discovery on those hits to try to get a compound that can move into the clinic. And the company then has opt-in rights at several different places, either at lead series or candidate selection, with predetermined milestones and royalties for moving forward. And so this partnership with System XC was really, just go back, it took us about, we selected the target with them. It took us about a year to set up this assay development and optimization. They then screened, and then it took us about 2 years to come up with good looking System XC inhibitors. And so ultimately, we were able to find a lead series of System XC inhibitors that weren't micromolar. These are nanomolar potent compounds. Their potency correlated with inhibition of glutamate release, and they look like drugs. They were orally bioavailable in multiple species. We saw good brain penetration, brain to plasma ratio, almost of unity. So these look like real drugs that could move forward. And this ultimately led to AZI licensing these series of compounds for clinical development. And they're currently active with this project at AZI. And so this is just some press releases on that collaboration and partnership. But hopefully this vignette one gives you a sense of how pharma and academia can work together in a kind of win-win, mutually beneficial way to advance a drug discovery project. All right, so that ends vignette number 1, and now I'm going to move on to vignette number 2. So vignette number 2, I think, is an example of how an academic collaborations, 2 different academic labs with different expertise can work together. And in this case, take something from a basic idea, from a bench, a discovery, all the way to the bedside. And this project is a collaboration between Johns Hopkins Drug Discovery, our chemistry colleagues at IOCB in Prague, and then the Bloomberg Kimmel Institute for Cancer Immunotherapy at Hopkins with Bob Leone and Jonathan Powell. So this was a real team effort, academic collaboration, which enabled us to move this glutamine antagonist project forward into the clinic. So let me again give a little bit of background on the project, and then tell you what we've done from a drug discovery perspective. Okay, so first, this project is focused on this drug called DON, which is 6-diazo-5-oxonorleucine. But from now, I'm just going to call it DON. And this DON is a broad and competitive glutamine antagonist. And so what I'm showing here, this is the structure of glutamine, and this is the structure of DON. And you can see they're identical, other than glutamine has an amino group here where DON has this active diazo warhead. So DON binds to and covalently inhibits glutamine utilizing enzymes. So DON is a natural product. It was identified back in the 1950s from bacteria in Peruvian soil, and was shown to broadly inhibit, specifically, but broadly inhibit glutamine utilizing enzymes. And what I mean by specific is it specifically inhibits multiple enzymes that use glutamine. And so, and it's known that glutamine is critical for tumor growth. And because Don inhibits glutamine, it was shown that Don could have a very strong inhibition of tumor growth in multiple preclinical models. And so here I'm just showing you the utility of glutamine in multiple cycles within a cell. Glutamine is involved in nucleotide synthesis, coenzyme synthesis, the TCA cycle, and glutaminolysis, amino acid synthesis, hexosamine synthesis. So it's very active within the cell. And Don inhibits all of these. So Don it inhibits all of these glutamine utilizing enzymes and processes. And so again, because glutamine is critical for tumor growth and metabolism, Don was shown, because it inhibits the utilization of glutamine, to be robustly effective in inhibiting tumor growth in multiple preclinical studies. And ultimately, this was taken into the clinic based on this active preclinical data. And then Don, and this is back in the 1960s and 1970s, there was about a dozen clinical trials, phase 1 and phase 2 trials in cancer patients, showing that Don, if given to patients, had strong anti-tumor activities in many different cancer clinical situations. And I'm just showing you a couple of the publications here. Unfortunately, though, Don wasn't tolerated very well, and so its development was halted. And when I say it wasn't tolerated very well, the main toxicity was GI. So when patients were taking Don at doses that were inhibiting their tumor, they were getting nausea and vomiting and gastritis. And because of this, their development was halted. The development of Don was halted. So we got interested in this and thought that perhaps we could use Don. We knew it was active, but we knew it was toxic. So our idea was, could we create a prodrug of Don? Okay, prodrugs are just an active compound that has more chemistry on it, making it inert until it gets bioactivated in vivo. And so what our idea was, could we create a prodrug of Don, which is preferentially activated in the tumor, right, and to Don, but less activated within the gut. And that way we could retain this therapeutic activity, but not have the same amount of GI side effects, and thus be able to go back into the clinic with this glutamine antagonist therapy. So that was basically the thought that we had, the strategy we had. And so here's just a schematic of that. So the idea was we wanted to use pro-moieties on Don that would be preferentially cleaved off by enzymes that are enriched in the tumor. So here's Don, very active. We stick a pro-moiety on it, and then it's not active. It's going to be inert, and it's going to circulate as an inactive prodrug. And the idea was that we would put these pro-moieties on Don in such a way that it would be cleaved off by enzymes that are preferentially enriched in the tumor. Okay, see, these are enzymes like cathepsins and HDACs and plasmin that are known to be quite abundant and prevalent in tumor. So the idea is we give Don, and it would circulate inactive, but when it got in the presence of the tumor, it would be activated to Don. So that was the basic idea. And so an ideal prodrug would be a prodrug that was stable in plasma, right? We don't want it to form Don in the plasma. We want it to be stable in liver. We wanted this prodrug to be stable in the gut. Very important to be stable in the gut, right? That's where Don is most toxic. But we wanted it to be preferentially cleaved. These pro-moieties preferentially cleaved to liberate Don in the tumor environment. And so we made hundreds of tumor-targeted, what we strategized would be tumor-targeted Don prodrugs. We synthesized hundreds of them. This was really a chemistry effort led by Pablo Mayer and Lucas Tenora at IOCB. And again, I'm not going to bore you with all the chemistry of making these, but I'm just going to go to the ones that worked and show you some examples of the most successful prodrugs that we made. And so the example I'm going to give is these tumor-targeted prodrugs that we made using acetylated lysine pro-moieties. And again, just a simple schematic of how this works. So here's our drug, in this case, Don, and we put acetylated lysine pro-moiety on it. Okay, when the prodrug's on it, it's inactive. The idea was that the acetyl group would be cleaved by HDAC, which is enriched in the tumor microenvironment, clipped off the acetyl group. And then cathepsin L, again, enriched in the tumor microenvironment, would clip off the lysine, and we would generate DON in the tumor microenvironment. And here's just the structure of that acetylated lysine, HDAC, and then cathepsin L cleaving and liberating DON. Now, this was not our idea. We took this idea from a publication that Michael Heyman had published looking at selective cancer targeting with another series of cancer drugs. But they showed that this HDAC cathepsin L acetylated lysine pro-moiety was able to direct the drug to the tumor. And so we then made several of these, we made probably about 50 different analogs with this acetylated lysine and other pro-moieties that aided in tumor uptake. Again, I'm not going to go through the chemistry, but just show you the best ones. So this is a compound called JHU 607, structure shown here. And what we found was what we were looking for. We found the compound was stable in plasma, so it didn't generate DON in plasma. It was stable in liver. It was stable in GI, very important, stable in GI tissues. It was not delivering DON, but in tumor, it was unstable. And when we looked at tumor cells that were in plasma, what we see is within plasma, it stays as the intact prodrug, very little DON liberated. However, when we look inside the tumor, what we see is very little prodrug, all of the DON is being liberated. So in vitro, we're seeing the profile that we want. Stable in plasma, stable in liver, stable in intestine, but delivering DON preferentially to tumor. So this positive in vitro data led us to then go in vivo. And so this is a mouse with a EL4 tumor, and we gave them the JHU 607, and we looked for DON. Where is DON? What we found was that there was about 5-fold higher DON in the tumor than there was in plasma. And probably most critically, when we looked at tumor versus gut, again, that where we want the compound in the tumor, where we don't want compound in the gut, we see a 12-fold difference. More DON that's in the tumor, very little DON generated in the gut. And so this was super exciting. This was showing us that we could take this drug we knew was active but toxic, and we could make prodrugs of it to deliver it to where we wanted it to go, but keep it out of the GI tract where it was causing toxicity. And based on this data, and this was work led by Rana Race in the lab and published here in medicinal chemistry, Journal of Medicinal Chemistry. But this data enabled us to then put together a pitch deck and talk to investors, and ultimately raised over $40 million and started this Drayson Pharmaceuticals to take these compounds into clinical development in cancer patients. And so we started Drayson Pharmaceuticals, and of course the people at Drayson wanted to pick their own compound, so they picked a different compound, which they then renamed DRP 104. And this compound was selected to move into the clinic. And like the compound I showed you before, it delivered much more DON to the tumor versus the gut. So this is the same experiment I showed you before, but a different drug. And we're showing that DON levels are 6-fold higher in tumor than plasma. And then when we look at tumor versus gut, we see an 11-fold. So we're getting more DON delivery, 11-fold higher to the tumor than we are to the gut. This was in mice. We wanted to see if this also happened in other species. This was kind of cool. We collaborated with the Colorado State Vet School. So in the vet school, people were bringing in their pets. These are mostly Labrador retrievers that have surface lymphoma. And what we did with these pets is we gave them our drug and then took plasma and then biopsied tumor. And what we found was, just like in the mice, in plasma, we see mostly the intact prodrug, not much DON. And when we sampled the tumor, we saw a 5-fold higher level of DON in the tumor than we did in plasma. So we're seeing very similar to the profile we saw in mice, also in a second species, which was quite encouraging. one of the really interesting, I'd like to say we designed it this way, but we didn't. We realized that what was happening with this DRP 104 in vivo is that it was actually bioactivating to DON in the tumor, but actually inactivating itself in the gut. And this is really quite interesting metabolism here. So here's DRP 104. It has this isopropyl ester and this acetylated tryptophan. And what was happening in the tumor is we initially saw this amide hydrolysis. So this acetylated tryptophan was coming off, and then the isopropyl ester was spontaneously coming off, and it was bioactivating to DON. In contrast, when we looked at DRP 104 metabolism in the gut, what we saw was this ester was coming off first, and then it was a dead end. What we saw is we generated this, after the ester came, this isopropyl ester came off, we were left with this compound, which we called the M1 metabolite. But this M1 metabolite was stable and inactive, and it never delivered DON. This was a dead end. And it was this difference in activation in the tumor and inactivation in the gut that was giving us this large separation of the generation of DON. And this, given this, what we saw, which I guess one would expect, is that we saw DRP 104 was similarly active to DON from an efficacy standpoint. So equimolar DRP 104 versus DON, they were both tremendously and robustly active, but DRP 104 was not toxic and DON was toxic. And so it's showing here, this is a tumor growth in a mouse. So these animals treated with vehicle, they have huge tumors. If they were treated with DON or DRP 104, robust inhibition, the tumor's completely gone. However, if you look at the weight change, the animals that were given DON lost like 30% of their body weight and had to be sacrificed. Now, they didn't have any tumors, but they died because of their significant toxicity. So when you look at overall survival, the animals that got vehicle or DON both died in the course of this experiment, while the DRP 104 had 100% survival. And again, this is because of the difference in toxicity, mostly to the gut. And here's just pictures of the gut from these animals. Here's a normal animal. You can see here's this animal that's been treated with DON, a complete loss of the crypts, severe ulcerations, lots of neutrophils and lymphocytes that have come in, versus DRP 104, which maybe doesn't look normal, but looks way better than DON. And so again, we created this prodrug that could get active to tumor, but not to the gut, which leads to the same efficacy, but a ability to get rid of the GI toxicity, which is allowing us to go back into the clinic. And so just this past year, this was published really as the first prodrug, I think, that's ever been described, which can bioactivate in one tissue, but bioinactivate in another tissue. And this was published in Science Advances this past year. In addition, and I didn't have time to talk about this, but I think it's really exciting. In our collaboration with Bob Leone and Jonathan Powell, we also discovered a completely novel mechanism of glutamine antagonists. And what they showed was that these glutamine antagonists induced a different metabolic effect in tumors versus immune cells. And this is really interesting. These same compounds could kill the tumor, but they could activate the immune cells. And this is because the cells used a different way to get around the tumor, glutamine antagonists that the tumor cells couldn't. So this led to a science publication, ultimately New England Journal of Medicine, did an article about how exciting this was from a therapeutic perspective. And so I think hopefully this gives you an example of an academic collaboration between two labs with really different expertise that could take an idea from bench to bedside. And this is from Drayson Pharmaceuticals website, and we were granted fast track designation of this DRP 104 for non-small cell lung cancer. And okay, so that's the end of vignette 2, and I'll end now with the last drug discovery example. And this is a newer one in our lab, and really like this one. This is an example of a drug discovery program where we turned a failure into success. And this is looking at GCP2 inhibition for the treatment of inflammatory bowel disease. And this was a collaboration we had with the Crohn's and Colitis Foundation. And so again, a little bit of background. This is an enzyme target called glutamate carboxypeptidase, or GCP2. In the brain, this enzyme is known to regulate glutamate transmission. In the gut, it also regulates glutamate transmission within the enteric nervous system, and also thought to be involved in folate metabolism. Folate has this polyglue tail on it, and this enzyme is thought to clip off those polyglue tail, so that results in reduced folate that can be taken into cells. This is also highly abundant in prostate, specifically in prostate cancer, but it's completely unknown, its mechanism there. So my lab has been interested in inhibiting GCP2 for brain disease forever. In fact, this was my thesis project, was identifying this enzyme. So this is a very old project in our lab. And it's well known that this enzyme, too much of GCP2, can cause excess glutamate transmission. It's implicated in lots of different neurological and psychiatric disease, several different pain states, epilepsy, seizure, drug addiction, schizophrenia, ALS, and the like. And actually in the literature, there's probably over 50 different animal models of disease by multiple labs showing that if you give an animal a GCP2 inhibitor, you can limit glutamate and have therapeutic benefit in these models of neurological disease. So that's the good news. So because of this, we've synthesized thousands of GCP2 inhibitors, and most of them are very potent, picomolar to nanomolar affinity, and super selective. I'm just showing some various classes of these GCP2 inhibitors that have been made. They're all have a zinc chelating group like phosphonate or urea thiol hydroxamate linked to this glutamate mimic that acts at the glutamate recognition site of the enzyme. And so the good news is these compounds are super potent and super selective. The bad news is that they're all polar and they're ionized at physiological pH. They don't get in the brain. They have poor oral bioavailability and really poor brain penetration. And because of this, no GCP2 inhibitor has ever been developed for CNS disease. And so we gave up. The lab gave up probably 8 years ago. We said, all right, we tried enough. We haven't been successful. We're gonna stop. And just about the same time where we were giving up, there was this paper showing that this same enzyme was highly upregulated in patients that had IBD. And this was the first paper that came out in PLOS ONE showing in Crohn's disease, this gene signature showed a huge upregulation of GCP2, which was correlated to many of the other IBD genes. And here's just a picture from that paper showing brown is where GCP2 is. So here's the epithelium of the ileum, and you can see normal versus IBD, huge difference. And then this now has been replicated by I think 6 or 7 different independent labs in different ways in different IBD stats. So this is a very significant finding that has been replicated by multiple labs. And so we got interested in this. We thought, oh, all right, we can't get our drugs to the brain, but we can get them to the gut, right? That's much easier. And so we took one of our tool compounds, 2-PMPA, and we worked with 2 different labs to see if it would work in colitis models. This was both Laura Enzyme and Shuang Li at Hopkins. And we found that this tool compound could reduce colitis severity. If you're a mouse, it was definitely helping the colitis disease in models of DSS colitis, IL-10 knockouts, and TNBS colitis. But 2-PMPA you can buy from Tocris, right? So it's not an ideal drug. We don't have any patent position on it. It's got low permeability. It required pretty high doses to work, and it's not orally available. And so we then took this idea to the Crohn's and Colitis Foundation, and in partnership, we discovered an orally active, gut-restricted, and proprietary GCP2 inhibitor. And again, I'm not going to go into the chemistry. We made about 100 different GCP2 inhibitor drugs and conjugates. This work, again, was led by Dr. Pablo Mayer and Rana Reis. We were looking for a drug that was potent, it was gut restricted, orally active, and well tolerated. And again, I'm just going to show you the winner. So this compound that we call SIBD3540 was chosen now as the clinical lead. I'm showing the structure here. It's deoxycholic acid linked to 2-PMPA. It's a super potent inhibitor for nanomolar potency. It's gut restricted. So when we give this orally, if we give this orally, we have 50-fold enriched drug in the GI system versus plasma. And we've now done non-GLP tox dose ranging in 2 species, and we have not seen any tox associated with this in the GI or systemically. And so this compound we've chose as the lead. And Diane Peters in the lab showed that this compound is quite efficacious in murine models of colitis. And in fact, when we look at it versus sulfasalazine or tofacitinib, a JAK inhibitor that's used in colitis, we see better activity. Again, this is a mouse model, but in the mouse model, we see better activity versus the used doses for sulfasalazine and tofacitinib. This is a disease activity index. You give the animal DSS, they get very sick. High numbers are worse. And what you can see is that our compound administration of this IBD 3540 is lessening the disease severity. Here's versus tofa, and here's versus sulfasalazine, and you can see in both cases we have superior efficacy, which is, it was quite exciting. And so also at the point that we're getting efficacy, we are able to bring down this elevated GCP2 activity in the gut about 80%. So we're getting really significant target engagement here in the gut, being able to normalize this aberrant activity. And this just last month, 6 weeks ago or so, this was published in Science Translational Medicine as the discovery of this compound for colitis. And just the last part of this, which is brand new and not published, is that I'd mentioned initially that we had been interested in GCP2 inhibitors in brain for neurological disorders. One of the areas that we had studied was pain. We had looked at this in neuropathic pain, peripheral neuropathy, chemo-induced pain, diabetic neuropathy-associated pain, inflammatory pain, cancer bone pain, and seen very good activity. So the question we ask is, could this be analgesic in abdominal pain? Because that's a pretty significant unmet medical need. And so we worked with Dr. Jay Pastrika at Hopkins, who has a chronic abdominal pain model. It's a TNBS enema model where he implants electrodes and can do a colorectal distension and looks at visceral motor responses. Animals that have gotten TNBS have a more extreme reaction to the colorectal distension than normal animals. And what he showed in this model was that we completely normalized this abdominal pain hypersensitivity. So here's the visceral motor response in blue, which is normal animals. Here's the animals that have TNBS. They have a hyper response. And if you give either gabapentin, 300 milligrams per kilogram, and that can normalize it. And we also showed that 10 and 1 could completely normalize this abdominal pain response. We're now doing lower doses since we didn't get to the minimal effective dose. And so I think this is my last slide. I think what's really cool here is I think we're in a very unique position with this drug because we have a single drug that can both inhibit IBD as well as inhibit abdominal pain. And this was just partnered with Blackbird Labs. Maybe many of you know this, maybe some don't. And Blackbird Labs is a brand new biopharma innovation hub that was started in Baltimore just this year from a $100 million grant from the Stephen Renee Bisciotti Foundation. Blackbird is homage to the Baltimore Ravens, since Bisciotti owns the Baltimore Ravens. But this is a group that's helping academic investigators basically translate their academic research into a company and to help move this into the clinic. And I think we're the first project that they are supporting to help move. So this is exciting, and hopefully we'll stay in Baltimore to move this company forward. And so I think that's my last slide. Hopefully I told you a little bit about Johns Hopkins, the fact that there's a rising role that academia is playing in drug discovery, and hopefully give you 3 examples of academic drug discovery vignettes. And then just to add with a thank you to the lab, this is my group at Johns Hopkins Drugs Discovery, and to thank all the funding sources from NIH and disease foundations and biopharma. And I'll stop there and take any questions.
All right. Thank you so much, Dr. Slusher. That was really wonderful. Really appreciate you coming and speaking to us on this. Does anyone have any questions? You can either put them in the chat or in the Q&A section. I may have seen a hand, but it disappeared. So. Thank you from Susan Dorsey.
Must have been perfectly clear.
Yeah, exactly. Great. All right. Well, if no one else has anything, thank you all very much for coming. Just as a reminder, again, next week is the annual Cockman lecture from Dr. Stone on IgG4 related disease. So we will see you all in a week. Thank you again.
Thank you.